Hello, welcome to the on-demand ProteOn webinar series. This webinar will show you the recent novel applications of the ProteOn XPR36 system and useful techniques. My name is Ruben Luo, and I am the Global Product Manager of the ProteOn XPR36 product line in Bio-Rad laboratories. This presentation will first overview the XPR36 technology, and then we'll go through the novel applications and useful techniques. The ProteOn XPR36 system is a SPR or surface plasmon resonance-based biosensor for biomolecular interaction analysis. And the XPR36 technology is a novel design of fluidics to enhance the performance of the SPR biosensor. And to illustrate the way SPR works, let's make an example. Say if you have a pair of molecules that you want to study their interaction. So using the SPR technology, you can first immobilize the first molecule to a sensing surface. For example, here, you activate a sensor chip surface and immobilize the first molecule. In SPR, we call it ligand to the surface, and then deactivate the surface. This is the first step. And the second step is you flow over the other molecule, and in SPR, we call it analyte. So the analyte will interact with the ligand. The binding phase we call association. And then when we inject running buffer, you will observe the dissociation of the analyte ligand complex. So this phase is called dissociation. And then the association and the dissociation provide you the information of the interaction between the two molecules. You can further regenerate the surface to remove all the analyte and repeat the interaction analysis. The SPR technology is able to provide rich information about the interaction between biomolecule A and biomolecule B. And usually, the user of SPR obtains the specificity of the interaction and the kinetic constants Ka and Kd, as well as the equilibrium constant, the big Kd. So they are the parameters describing the properties of these two biomolecules and their interaction. And as we listed here, almost all types of biomolecular interactions can be analyzed using the SPR technology. And the ProteOn XPR36 system, as I mentioned, it incorporates a novel fluidic design so that the performance of the SPR analysis is enhanced. So the system comprises an instrument and software plus the consumables. And with the consumables, especially sensor chips, we are able to provide multiple surface chemistries for your particular applications. Basically, the novel design of fluidics in the XPR36 technology means the 6x6 interaction array. So let me illustrate the interaction array using the following animation. So in the XPR36 technology, you first activate the 6 channels in parallel. So we have a fluidic module to create 6 channels on the sensor chip surface. And then you can immobilize 6 different ligands, or one ligand at 6 different conditions to the surface, into the vertical channels. And then you can deactivate these 6 channels. And then the important part is here, you rotate the fluidic module to create 6 horizontal channels after the ligand immobilization. And then you can inject 6 different analytes or one analyte at 6 different concentrations, and to monitor their interaction. And then you can rinse the surface to measure the dissociation. So by this means, one injection, you have 36 different interactions, and you can monitor 36 different sensograms just using one injection in the ProteOn XPR36 system. The advantages of this 6x6 interaction array are apparent. First, the versatility, which means you can use the interaction array to design different types of experiments for your particular purposes. For example, experimental optimization is essential for SPR experiments. So you can use the 6x6 array to test the different conditions of the ligand and different conditions of the analyte. Just by one injection, you can test 36 combinations of the conditions and find the best one for your interaction analysis. People also use this array for high throughput screening because you can immobilize 6 different ligands to the surface and inject one analyte at 6 different concentrations. So we call it kinetic screening, because you obtain 6 full kinetic data sets in one injection. And you can also use it for multiplexed screening and even to create a 36 different ligand array and use it for high throughput screening. The second advantage of the XPR36 technology is data quality. Because we have multiple channels in parallel and they all run in a simultaneous manner, so you can use one channel as a real-time referencing channel. So you can use it to correct the baseline drift when you use capture surface. Capture surface means you have a covalently bound capture reagent on the sensor chip surface, and the capture reagent captures the ligand for interaction analysis. So in this case, baseline drift is inevitable in SPR experiments, and the real-time referencing provides you a perfect correction of the baseline drift. And the capture surface is widely used in drug discovery. The third advantage is productivity. Since we have 6x6 interaction array, which provides you 36 sensograms at one time, the data production is faster compared to traditional SPR biosensors. Okay, now let's talk about the recent novel applications that have been applied on the ProteOn XPR36 system. And we will go through the fragment-based lead discovery, cell-antibody interaction analysis, and a novel drug delivery model. Fragment-based lead discovery or fragment screening is a popular methodology for the discovery of small molecule drugs. People employ different technologies, including SPR, to screen small molecule pieces, which are called fragments, and find positive hits. And then these positive hits can be used to build scaffolds, which means larger small molecules, and test again using the same drug target. And then you find the positive hits from the scaffolds and use them to build lead compounds. So this is a bottom-up process to build your drug candidates. And of course, in the screening process, the binding affinity will increase from fragments to scaffolds then to lead drug compounds. The SPR is a mainstream method to measure the affinity between the small molecules and drug targets. This is an example of fragment screening using the ProteOn XPR36 system that we obtained together with a collaborator in UK. The project is screening fragments against a DNA repair protein. As shown here, the screening sensograms were successfully obtained, and the kinetic analysis between the fragments and the target protein could be well obtained. And this work has been published in the Bio-Rad journal Bioradiations, the October issue. And we have another webinar delivered earlier this year by Dr. Alan Doliso from Australia, also focusing on his excellent work of fragment screening using the ProteOn XPR36 system. If you want to view that webinar, please contact us. Another novel application is using the ProteOn XPR36 system to analyze the cell-antibody interactions. And we have multiple works achieved by our Japanese ProteOn XPR36 users. And there was an article published in Japan, and if you need further information, please contact us for the article. And the workflow is first you immobilize the antibody on the surface, and then you inject cells as analytes to measure the interaction between the antibody and the cell. So in this case, you can measure the interaction between your reagent and the native membrane proteins on the cell membranes. This is a proof of concept of the cell-antibody interaction analysis. So in this case, we mobilized an antibody on the surface and flow over the Staphylococcus aureus cells as analytes. As we know, this bacterial cell has protein A on its surface. So SPR provides the interaction analysis between the whole bacterial cells and the antibody. So as shown here, the binding analysis was achieved using the ProteOn XPR36 system. And as I mentioned, the article published in Japan contained more examples between antibodies and mammalian cells. The third novel application is a drug delivery model. It is based on a reagent kit we launched last year. It is called Liposome Capturing Kit, or LCP kit. Using this kit, we are able to immobilize liposomes on the sensor chip surface and measure the interactions between small molecule drugs and liposomes. This methodology provides you the knowledge of penetration capability of these small molecule drugs through lipid bilayer membranes. And this is very important for you to understand the drug delivery of these small molecule drugs. We have another on-demand webinar focusing on this application, and you may view it using the links provided. So in the experiment design, we immobilized different liposomes in the vertical channels and inject the small molecule drugs in the horizontal channels. So this is a high throughput manner to analyze the interactions between the small molecules and the liposomes. And this slide illustrates the result of the interaction analysis. And as listed here, you see the different small molecule drugs and different liposomes. They have diversified interaction affinities. And also the dissociation shows you the retain of the small molecule drugs in the liposome. And it depends on both the small molecule and the liposome properties. And you are able to use this model for quantitative analysis of the binding affinity and penetration capability of the small molecule drugs with the liposomes. Okay, after the recent novel applications, we will talk about a few novel techniques on the ProteOn XPR36 system. The first two techniques, Quantikinetics and antibiotics detection, are based on the quantitation capability of SPR biosensors. And then we'll talk about regenerable Biotin-Capture surface. It's a novel surface chemistry based on our HTG and HTE sensor chips. And then the SPR-MS Coupling. This is preliminary study to couple the ProteOn XPR36 system with multi-mass spectrometry. SPR biosensors can be used for sample quantitation. And this quantitation method is highly efficient. In the SPR way, once you have a capture surface constructed that can be used to capture the target of quantitation, then you can quantify your samples very quickly because we measure the initial binding slope of the sensorgrams between the target of quantitation and the capture surface. And the concentration of the sample is proportional to the initial binding slope. So once you have a capture surface constructed, you can achieve quantitation usually within 1 minute or even within half a minute. This is an example of using the ProteOn XPR36 system for sample quantitation. Basically, you need to inject standards first to construct the standard curve, and then you inject your unknown samples to quantify the sample concentration. But since we have multiple channels in parallel, you are also able to inject your standards together with an unknown sample to quantify them using one injection. Quantikinetics is a technique created to combine the sample quantitation and the kinetic analysis in one workflow. It is designed for antibody analysis using the ProteOn XPR36 system. As we know, when we analyze antibodies using SPR biosensors, it is very typical to use capture surface chemistry, which means covalently linking a capture reagent such as protein A, protein G, or anti-Fc antibody to the sensor chip surface and use this capture reagent to capture the antibody samples. And then the next step, we inject antigens to measure the kinetics between the antibody and the antigen. And the first step usually is not quantified because it's just for the purpose of capturing the antibody to the sensor chip surface. And the second step, of course, is quantified for kinetic analysis. However, if you construct the capture surface well, then you will be able to quantify the antibody capture step using the ProteOn XPR36 system. And this step will provide you the information of the antibody sample concentration. So within the first step, you not only capture the antibody samples to the sensor chip surface, you also quantify the antibody samples. And then in the next step, you inject antigens to measure the interaction for kinetics. So the first step is constructing the capture surface, and the second step, we capture the antibody to the surface while quantifying these antibody samples. And the third step, we measure the kinetics between the antibodies and antigens. So these is the whole workflow of the Quantikinetics technique. And as we show here in this table, we were able to use the ProteOn XPR36 for quantitation of antibody samples in a wide range, and they all work pretty well. And the measurement time for each cycle, one cycle means quantifying 6 samples, and then for kinetic analysis is pretty short. As we see, usually within 1 hour, you can complete the quantitation and kinetic analysis of 6 antibody samples it is very efficient. And the effective range on the ProteOn XPR36 system is broader than the usually than typical ELISA assays. Another technique based on the quantitation capability of the ProteOn XPR36 system is to detect antibiotics in food, for example, milk. This is an interesting topic because food safety has become a significant topic in certain countries, and SPR could be a novel method complementary to the current mainstream LC-MS or immunoassay methods. So in SPR, we could design a competitive immunoassay to detect these small molecule antibiotics. And what we do is, as opposed to the traditional SPR configuration in protein small molecule analysis, which is covalently linking the large molecule to the surface and flow over small molecules. In this case, we immobilized the small molecule antibiotics to the sensor chip surface and flow over the antibody that was raised against this small molecule antibiotics. And in this manner, we are able to run the competitive immunoassay because we can mix the specific antibody with the food sample. And if the food sample has the same antibiotics in the sample, then the antibodies will be saturated. So you will not see a binding of the specific antibody to the antibiotics on the sensor chip surface. Otherwise, you will see a strong signal from the binding of the antibody samples because these antibodies are not saturated by the antibiotics in the food sample, and since it's a large molecule, it will generate significant signal in SPR experiments. As a proof of concept, we detected spiked milk samples. So we spiked the milk samples with antibiotics called tyrosine, which is commonly used in cattle, such as cows. The small molecule tyrosine has an aldehyde group, so we were able to immobilize tyrosine to the sensor chip surface through aldehyde coupling. So as illustrated in this graph, we first modified the sensor chip surface with EDC NHS and carbohydride, and then we linked the aldehyde group to the surface using shift-space reaction, and then we reduced the carbon-nitrogen double bond to a single bond so that the tyrosine molecule is covalently permanently linked to the sensor chip surface. So after the immobilization of the antibiotics, we tested the uniformity of different channels. As I mentioned, the ProteOn XPR36 system has 6 channels in parallel, and you inject samples simultaneously in all these 6 channels. So we detect the uniformity of the 6 analyte channels, and they showed very nice consistency. And then we created standard curve using prepared samples. So these are the samples we prepared with tyrosine, just in running buffer. And then we tested the spiked samples. So spiked samples means the tyrosine added milk, and we treated the milk, extract the supernatant, and then mixed with the antibody, dilute it in running buffer, and inject into the system. And we were able to quantify the antibiotics tyrosine and the spiked milk samples. And these 2 curves showing the standards and the spiked samples. So the spiked milk samples, they showed very consistent data with the standard samples prepared in running buffer. So the consistency between these 2 curves showed us that we have the capability to detect the tyrosine concentration in real milk samples. Okay, let us move to the next technique. We call it Regenerable Biotin-Capture surface. As we know, Biotin-Capture is the very commonly used capture method in SPR experiments. It means you covalently link streptavidin or neutralavidin to your sensor chip surface, and then use streptavidin or neutralavidin to capture biotin-related ligand samples for the following interaction analysis with the analytes. The problem with this surface chemistry is that the binding between the biotin-related ligand and streptavidin or neutralavidin is too strong, so you cannot remove the ligand from the surface. You have to dispose the sensor chip after the analysis unless you want to use the same ligand again. But we were able to create a novel workflow to facilitate the regeneration of the chip surface so that you can immobilize different ligands, different biotin-related ligands to the surface on the same sensor chip. So what we did was we used the ProteOn HTG and HTE sensor chips, which contain tris-NTA surface chemistry. And the tris-NTA surface can very strongly bind his-tagged proteins so that you will not be affected by the baseline drift normally you observed with his-tag captured surface. So in this case, as I illustrated in this graph, the tris-NTA surface, we call it, these tris-NTA groups, we call them the secondary capture reagent. And we use his-tag streptavidin as the primary capture reagent. So we first capture the his-tagged streptavidin molecules by the tris-NTA groups on the sensor chip surface. And then we use the his-tagged streptavidin to capture the biotin-related ligand for interaction analysis with the analytes. And after the SPR analysis is completed, we inject regeneration buffer solutions to remove all the his-tagged streptavidin, ligands, and analytes from the surface. And the sensor chip surface will be restored. So it will contain only tris-NTA surface. And then you are able to go to next cycle to immobilize his-tagged streptavidin again, and then new ligand for new analyte. And these will give you the chance to run multiple interaction analysis using the same sensor chip. And it's very useful when cost is a concern in designing SPR experiments. And we have another on-demand webinar focusing on this application, and you may view it by clicking one of the links. So this sensorgram shows the whole workflow. So first, we activated the HTG or HDE sensor chip surface with nickel sulfate solution, and then we captured the his-tag streptavidin, and then we used this his-tag streptavidin to capture the ligand, which is biotin-related anti-rabbit IgG antibody, and then we measure the interaction between the ligand and the analyte. The analyte is rabbit IgG antibody, and then we regenerate the surface to remove the his-tag streptavidin, the ligand, and analyte, so you are able to go to the next cycle of interaction analysis. These sensorgrams present the repeats of the same interaction analysis in 3 consecutive cycles. So as you see here, the sensorgrams from experiment 1 to 3, they are all consistent, which means we were able to effectively regenerate the sensor chip surface between the cycles. And the regeneration had high performance so that the sensorgrams are consistent between the different cycles. And this table shows the kinetic constants and equilibrium constants we obtained from the 3 experiments, which were from the 3 consecutive cycles. And these parameters are all consistent, which validates the workflow of this regenerable Biotin-Capture surface. Okay, the last but not the least technique is the SPR-MS spectrometry coupling. As we know, the SPR technology analyzes the interaction between two biomolecules. This is so-called characterization. Mass spec, as another powerful technology, identifies biomolecules in fine details. So if we can combine these two powerful technologies, we will be able to utilize the advantages from both sides and provide you more understanding of the biomolecules of interest. So in this case, we had our first attempt to combine the ProteOn XPR36 system with mass spec analysis. We analyzed the interaction between an antibody and the beta amyloid peptide fragments. So we first analyzed the kinetics between these two biomolecules, and we obtained the kinetic constant and the equilibrium constant. In the next step, we took the sensor chip out of the ProteOn XPR36 system, washed the sensor chip surface to remove the salts, and put a droplet of MALDI matrix on the surface. Once the surface was dry, we mounted the sensor chip prism in a customized adapter and inserted the adapter in a mass spec instrument for analysis. The analyte could be detected and identified using the mass spec analysis and MS fragment analysis, and then followed by mascot search. Okay, this is the end of this webinar presentation, and we really appreciate your attention and time. And if you have questions, please email me or type the questions in the Q&A box.